Hello. हेलो, आई एम ऑडिबल? मेरी आवाज आ रही है सबको? जल्दी जल्दी ज्वाइन करो, फिर मैं स्टार्ट करता हूँ। हॉरिजॉन्टल जीन ट्रांसफर्स का क्या इंपॉर्टेंस है बैक्टीरियल इवोल्यूशन में। बैक्टीरिया ने कैसे बहुत सारे जीन को हेलो, आई एम ऑडिबल ट्रांसफर किया। मेरी आवाज आ रही है सबको? जल्दी जल्दी ज्वाइन करो, फिर मैं स्टार्ट करता हूँ। हॉरिजॉन्टल जीन ट्रांसफर्स का क्या इंपॉर्टेंस है बैक्टीरियल इवोल्यूशन में। बैक्टीरिया ने बहुत सारे जीन को ट्रांसफर किया। So let us start today's topic about horizontal transfer and antibiotic resistance. How the gene is responsible for making antibiotic resistant, developing antibiotic resistant when there is a lot of infections. So this is related to the exactly we are focusing on this topic, how evolutionary, what is the evolutionary importance of this antibiotic resistant gene transfer and how horizontal gene is responsible for so many spreading of harmful genes in the microbes. So exactly what we can define horizontal gene transfer, a kind of lateral gene transfer. Do you know about the lateral gene transfer? Any idea about the lateral gene transfer or horizontal gene transfer? What is the difference? What is HGT, horizontal gene transfer? Exactly, if you're talking about the horizontal gene transfer, this is the non-sexual transfer of gene, where you can say direct transfer of gene from cell to cell. Direct transfer of gene from cell to cell, right? So that is the main mechanism of horizontal gene transfer, and later on by various experiment of Griffith and others, it was reported that there is a various mechanism of gene transfer. So exactly we can define what is the horizontal gene transfer. It refers to the ability or process of which piece of DNA is going to be transferred. So we will understand how this piece is going to be transferred and what are the gene responsible for that, what are the enzyme present in the bacteria during the transfer, how the cell is dividing, how the DNA is hopping from one cell to other. So if we're talking about the gene transfer, basically we can say it as a exchange of genetic material across one cell to another. And you can see there are three mechanisms of gene transfer exactly. One is the transformation, other is the conjugation, and third one is the transduction. And all three mechanisms is responsible for transfer of a specific gene from one cell to another. What is the difference between all three? We will understand if you'll see the basic mechanism by which antibiotic resistant gene is going to be transferred. This is a bacterial transformation, you can see. So bacterial transformation, what happens? There are two types of cells. One cell is donor cells, other is recipient cells. And basically, these two cells are contacting with each other. And if there is a breakage of donor cell and there is a release of large number of DNA, then you can see that recipient cell can accept these cells, fragment of DNA from one cell to another, from donor to recipient, right? So this is basically the uptake of DNA from one cell to another, and the donor cell is basically responsible for transferring the gene. So all the genes or all the DNA is not going to be transferred, rather than only specific gene is going to be transferred. And basically in the nature, you can see there are various mechanism may be operating, which is the cause of death of the cells, and that's why there is a release of DNA. And these DNA are going to be fragmented into the small pieces, very small pieces, mostly 10 kb or less than 10 kb. And these DNA are only specific few DNA are going to be uptaken by the recipient cell. That is the mechanism of bacterial transformation. Another mechanism is bacterial transaction. If you see the bacterial transaction, there is a phage-mediated genetic transfer. So bacteriophage, especially lambda phage, if you'll see, lambda phage is responsible for transfer of these bacterial gene accidentally from one cell to another. So we can say first a donor cell will be where phage is going to attack the E. coli cells, and accidentally during the lysogenian and lytic cycle, they are releasing these DNA, they are taking these DNA and then releasing these DNA into the recipient cell when they are injecting their own DNA. So that may be another mechanism by which fragment of gene can be transferred from donor cell to recipient. And third, what is the third mechanism? Excellent. The third mechanism is bacterial conjugation. If you see the bacterial conjugation, exactly what is happening in the bacterial conjugation? Bacterial conjugation, there is a formation of a specific contact organ that is called as the pilus. And a lot of question is going to be asked in the CSIR related to the conjugation and transformation especially. And also, in last few years, we have seen that the examiners have asked about the mechanism of spatial transaction. So we will study all the details, specifically about the genes, what are the different and how exclusively they can be transferred, especially the molecular detail we will study, because this is the microbial genetics, and this is one of the very important part of the examination. So if you'll see in summary, what is the difference between the transduction, conjugation, and transformation? So you can see transduction, basically the phage-mediated genetic transfer, you can say. This is the phage-mediated lambda phage especially, right? This is lambda phage involved in the transformation of the gene, and how they are going to be transform the gene. We can understand by the phage mechanism. Phage has a two type of cycle, lysogenic and lytic cycle. So lytic cycle is the death of the cycle where E. coli cell burst, and then this complete bacteriophage is going to be released in the environment. And when these bacteriophage, which is carrying the transfer gene, are attacking the next recipient cell, then they are going to release the same gene inside the bacterial cell, and they are going to be part of the E. coli genome, right? So that is called as the recombinant DNA. They are forming the recombinant DNA, and this is the way by which the gene is incorporated into the recipient cells. If you have any question, please ask. Don't hesitate. You can raise your hand and you can ask. Because if you know, you're appearing in certain exams like CSIR, so they are basically asking the practical aspects of the questions. And also there are a lot of interview phases when you are appearing for an interview in good institutes for PhD admissions. Okay, Gopal, okay, Adi. So, any question till now? Because this is interactive classes, you can interact, you can ask questions freely. There is no boundary, and we will try to answer all the things so that any doubts would not be remaining in your mind. Okay, so we'll see there's of pilus. Exactly, the pilus gene are there, and F plus and F minus. You might be knowing F plus is the kind of donor DNA, and F minus is the recipient DNA, and the gene is transferred by the pilus. We'll study later on the detailed mechanism, how the TRA gene, MOP gene are there, laxis gene are going to transfer the gene from the F plus to F minus. And if you're talking about the transformation, exactly, transformation is a different way of uptaking the DNA because here is the dead bacterium, and they are releasing lots of DNA in the environment, and these DNA going to be fragmented to pieces, and a competent cell, competent bacterial cell can uptake these kind of DNA. So you will see the transformation. There are natural kind of transformation, and there is artificial transformation. So in the laboratory, we are basically employing the artificial way of transforming the bacteria if you want to transfer the gene. This is the old technique. Nowadays, a lot of modern technique have been evolved, like gene gun method, which are basically involved in the making the competent cell. Competent cell अगर बनाना है आपको तो laboratory में बहुत सारे मेथड आजकल इवॉल्व हो चुके हैं, like gene gun, particle gun method. So they are basically helpful in rapid transfer of bacterium in the competent cells. And the later you can select those bacterium and you can check that really the cell has been transformed or not. All right. So these are the three basic mechanism of horizontal gene transfer, and all three are basically horizontal gene transfer: transformation, transaction, conjugation, and transformation. Right. So in one sentence, how we can define the conjugation? Conjugation by transfer of a gene from one bacterium to another by conjugation tube. And what is the transformation? This is the direct uptake of DNA, and transduction, one bacterium to another by viruses. So before studying about these mechanism, you must know the detail of the about the plasmid. What are the different type of plasmid and how they are going to be operative, right? And how they are acting. So what are their nature? What are the different type of plasmids? All these you must know about that. Okay. So some plasmid are conjugative, might be knowing, might have heard in the books. It has been mentioned, and your teacher might be taught in that way. And that conjugative plasmid have feature that they are self-transmissible. Other type of plasmids are there that are non-conjugative. They are difficult to transfer. They need help of some another genes by which they can transfer. Right. So conjugative plasmid harbor genes for the transfer machinery. Right. And if there are non-conjugative plasmid, they are mobilizable. If they can be transferred through conjugation when a helper transmissible plasmid is present. So they need always a helper plasmid. Okay. So I think this is clear. Any doubt is there? You can ask. No doubt? तो अगर किसी को डाउट है तो पूछ सकते हो. तीन तरह के प्लाज्मिड होते हैं. Conjugative एक तो होता है जो सेल्फ ट्रांसमिशन कर सकता है, and then conjugative plasmid harbor gene for the transfer machinery, and then non-conjugative plasmid that are mobilized, that can mobilize via some helper plasmid, right? The helper plasmid conjugate. Before starting all these things, let us start with some glossary points, like what is the phase lysate. So these are the suspension of released progeniphases outcome in the phase lysate, genetic mapping, and these are the generalized transduction method, which you can utilize for the gene transfer. Okay, so gene mapping, and you can prepare the linkage map. You can make a gene order. You can calculate the gene distance, map distance, and another kind of term often used in these microbial genetics is selectable markers. These marker genes are present over the cells, and where you can start the selection of transduced cells in the population. There are unselected marker, rest other marker are called as the unselected marker. And already we have discussed about the horizontal gene transfer. This is actually DNA hopping from one cell to another. What is the endozynode? Original genome of the recipient cell is called the endozynode. And the term, there is a exozynode, the DNA fragment transferred from donor cell to recipient cell is called as the exozynode. Right? What is the merozynode? These are the partial diploid cell, often can be asked in the examination, right? So you must know these terms. What are the transformation? Uptake of DNA fragment from surrounding cell wall. This is called as the transformation, right? This is called as the transformation. And another term is conjugation. Conjugation, unidirectional transfer of genetic material from donor to recipient bacterium through cell-to-cell contact. Okay, so just a minute. One minute, student. Just a minute. This is the first day we started these lectures. Hello, student. I'm audible. Check. So let us start again. So these are a few terms you must know, because these can be asked in your examination. So you must know these terms: endogenote, like exogenote, merogenote, transformation, production, prophase. Prophase is a kind of integrated DNA. Right? These are the kind of integrated DNA which can be used for the genetic transformation or the gene transfer in the bacterial chromosome. And another term I will know you will be using about the HFR, that is high frequency transformation. HFR, a plasmid is integrated on the chromosome, forming episome or recombinant DNA. Right? So trans okay. So I'll start about the conjugation. Conjugation, this is the transfer of genetic material from one bacteria to another. And this is by a bridge that is called as a conjugation tube. And this was exactly discovered by the Joshua Littleberg and Edward Tatum in 1946. Okay. And the first evidence for cell-to-cell contact was provided by Bernard Davis in 1950. And bacterial conjugation is also known as type 4 secretion system. So if you see the conjugation occurring between many species of bacteria, including gram-negative, gram-positive, and even the bacteria and plants. So if you'll see about F plus and F minus conjugation, because this is going to be asked, several combination can be asked in the examination. And so next time we'll study that. Thank you so much. Bye-bye.